Hello, everyone. Welcome back from the break to session 2 of the event. Couple of housekeeping things before we jump into the event. We've been getting a lot of emails about where to find the agenda. The agenda and the speaker bios can be found on our website, which is in the chat box and also on the screen that you're seeing right now. It's www.complexgenerics.org. You can also follow us on LinkedIn for any updates on this event and future events. We're pretty good at posting updates about slides being up on our website and any industry updates as well. So I would encourage you to follow us on LinkedIn if you'd like to receive any updates as soon as possible. With that, I'll hand it off to Andrew to kick off session 2.
Session 2 for mechanistic modeling of dermal generic drug products has 5 speakers today. In order of presentation, Khondoker alum PhD is a staff fellow at the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD. Dr. Alum's role in the division is to utilize translational tools such as PBPK modeling to address specific questions pertinent to drug development process and/or regulatory decision making. Priyanka Ghosh, PhD, is a senior pharmacologist at the Division of Therapeutics 1, Office of Research and Standards, OGD. Dr. Ghosh leads regulatory science research initiatives related to topical and transdermal drug products under the Gadufa Regulatory Science Program, and also leads the development of general and product-specific guidances and review strategies for preemptive meeting requests and citizen petitions for these products. Jessica Spires, PhD, is a senior scientist at Simulations Plus. Dr. Spires focuses on PBPK modeling of non-oral routes of administration, including dermal, ocular, and pulmonary administration in the GastroPlus and Membrane Plus software platforms. Sebastian Polak, PhD, is a senior scientific advisor in the SimCip division of Cetera UK. Dr. Polak is also the head of the mechanistic dermal modeling within SimCip. Eleftheria Tsakalozou, PhD, is a staff fellow at the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD. Her research interests include skin PBPK modeling, interactions between excipients and molecular targets, including gut transporters, and development of quantitative modeling simulation tools to support bioequivalence assessments. The moderators today are Raney and Arora, PhDs. Dr. Raney is the Associate Director for Science in the Office of Research and Standards, OGD. Dr. Raney is a thought leader in topical and transdermal drug products with over 30 years of experience in skin research, producing numerous research manuscripts, review articles, book chapters, and patents in pharmaceutical product development. Dr. Arora is a senior scientist in the biopharmaceutics group at Janssen. Dr. Arora's primary responsibility involves leading cross-functional teams to implement biopharmaceutical development strategies across preclinical development, clinical development, and CMC development.
Thank you for a nice introduction. My name is Khondoker. I'm a pharmacologist working in Division of Quantitative Methods and Modeling within Office of Research and Standards, Office of Generic Drugs. In my presentation today, I will provide an overview of our research activities, and then I will discuss about our regulatory experience on mechanistic modeling for generic dermatological drug products. Disclaimer of my talk today. In my talk today, I will be covering the opportunities of mechanistic physiologically based pharmacokinetic modeling or PBPK modeling for alternative bioequivalence approaches for generic dermatological drug products, our research roadmap towards developing mechanistic Dhammal PBPK models, and then I will be discussing the use of Dhammal PBPK modeling and simulation approaches in regulatory space. The agency publishes product-specific guidance on establishing bioequivalence for generic dermatological drug products. In vitro characterization-based bioequivalence approach can be adopted for many dermatological drug products. In general, this approach includes qualitative and quantitative sameness or no difference criteria, physicochemical or structural similarity or sameness, equivalent rate of drug release, and equivalent rate and extent of drug permeation using excised human skin. The agency often recommends in vivo study to demonstrate bioequivalence. The most common form of this kind of study is comparative clinical endpoint bioequivalence study. Other study includes BE study with peak endpoints, vasoconstrictor study, adhesion study, skin irritation, and sensitization study. Let's discuss about the challenges associated with comparative clinical endpoint bioequivalence study and how we can use in silico methodologies to overcome these challenges. This study may not be sensitive enough to detect formulation differences. We may see large variability in observed response. The study is relatively costly as it requires high number of subjects, and the entire drug development process could be lengthy. Model integrated virtual bioequivalence study can be a valuable option to overcome these challenges. The agency welcomes innovative approach given that the proposed approach satisfies requirements of applicable statutes and regulation. The advantage of Dhammal PBPK model is that we can estimate local tissue bioavailability for test and reference drug products. The model integrated virtual bioequivalence study can also be cost effective and may offer faster drug development process. In this slide, I will go over 3 important elements that we should consider during the development of Dhammal PBPK model. One of the most important component of Dhammal PBPK model is skin physiology. The physiology of the skin is fairly complex. It is composed of multiple layers such as stratum corneum, viable epidermis, dermis, subcutis. Each layer has its own characteristics property and defined thickness. The permeability and diffusibility of drug molecule of a drug product is governed by the interplay of the skin physiology with the physicochemical properties of API and formulation. So our mechanistic PBPK model should capture this interplay. The second important component of Dhammal PBPK model is the incorporation of formulation attributes. This is particularly important if the model is going to be used for virtual bioequivalence. Critical attributes of test and reference drug products such as biological properties, pH, specific gravities, physicochemical characteristics of drug product, and the potential metamorphosis event should be appropriately captured into the model. The third important component of the model is the parameterization process. To estimate or refine key model parameters, we can leverage the data from IPPT studies or other suitable form of studies such as skin biopsies, microdialysis, or systemic PK data. This slide will provide you a brief overview of workflow we can follow to develop the Dhammal PBPK model. The first step is to develop or decide model structure or modeling platform to be used for PBPK modeling. Then we can incorporate formulation attributes of drug product and physicochemical properties of API. Then we can choose the population type, for example, healthy or disease population, depending on the need of the PBPK modeling. We can utilize in vitro or ex vivo data to inform model parameters. The assessment of models performance is very important part in the overall approach of PBPK model development process. At some point, we may need to go back and refine certain model parameters, but this change has to be made with proper justification. Once we integrate all the important components of the model and model's performance is suitably verified, we can predict the disposition of a dermatological drug product in local tissues, systemic compartment, or other target site. In my next few slides, I will try to present our research roadmap towards developing mechanistic Dhammal PBPK model. Over the past few years, the agency has awarded many grants on Dhammal PBPK modeling. In 2014, we have awarded 2 grants. In 2018, 3 grants. In 2021, we will be awarding 2-3 more grants. Needless to say, all these grants were awarded with Gadufa fund. In these slides, I will be discussing our research objectives in more details. One of the objectives of our research activities is to improve our understanding on skin physiology in healthy and disease populations and develop and validate in silico healthy and disease skin model. Some of our grants have the objectives to characterize formulation attributes of selected semi-solid dermatological drug products and incorporate these key formulation attributes into the PBPK model to define a safe space for these quality attributes. Our grants also aim to capture the interplay between skin physiology and drug product quality attributes. We also emphasize on generating or collecting experimental and clinical data from in vitro, ex vivo, and in vivo methodologies Our objective is to validate these methodologies and use these data sets for PBPK model development and validation. Our ultimate goal is to develop a reliable in silico tool and establish in vitro, in vivo relationship for predicting unknown scenarios and performing virtual bioequivalence for topical drug products. This is a list of Gadufa funded research projects supporting the Dhammal PBPK modeling and simulation. Over the years, we have worked together with software companies, academia, and research institutes to achieve the critical mission of Gadufa. Please check out the link below if you are interested to learn more about our research activities. We are continuously working to improve the existing Dhammal PBPK model. A couple of grants has been awarded to Sartara to improve the Dhammal PBPK modeling platform within SimSip simulator. In these grants, Sartara and SimSip has collaborated with the University of Queensland and University of South Australia to generate necessary experimental data. We have also worked with Simulation Plus to develop the Dhammal PBPK model. Simulation Plus has collaborated with their industry partners to collect necessary data set. We have also awarded grant to improve open source PBPK modeling platform like PKSim. In this grant, the University of Waterloo and the Children's Hospital of Los Angeles collaborated together to achieve the grant objectives. We have further awarded grants to the University of South Australia, where the grantee collaborated with the Goethe University Frankfurt and the University of Surrey to make a specific improvement of Dhammal PBPK model. In all these grants, our scientists from the agency actively involved to provide technical insights which deemed critical to achieve the research objective of these grants. In this slide, I have listed some key outcomes we have achieved from our Gadufa funded research. One obvious outcome is we have developed mechanistic Dhammal PBPK model by incorporating skin physiology and formulation attributes. We have developed skin disease models. We have got IPPT modules where we can simulate the IPPT study results by incorporating experimental details. We have developed model to simulate vasodilation or constriction in the dermis following dermal application of certain topical drug products. We have improved the modeling platform to handle multi-site applications of topical drug products to resemble real-life scenarios or clinical trial situations. For example, a dermatological drug product may be applied to face and shoulders simultaneously. We have incorporated inter and intra-subject variabilities into PBPK model. This is very critical if the model is going to be used for virtual bioequivalence. Our ultimate goal was to improve the predictive power of Dhammal PBPK model so that the agency can have higher confidence on the model to be used for virtual bioequivalence. Now, let me give you a brief update on our research focus in fiscal year 21. This year, agency has posted 3 notice of funding opportunities to improve our existing Dhammal PBPK model or modeling platform. In RFA FD 21-019, we have asked for applications for developing a Dhammal PBPK model that can simultaneously take account the absorption and evaporation of vehicle or co-solvents. You may know that absorption of vehicle or co-solvents may change the skin physiology by disrupting the intercellular lipid bilayer of stratum corneum. This change in skin physiology may influence the permeability of API. On the other hand, the evaporation of vehicle may lead to metamorphosis of formulation. The objective of this grant was to capture both dynamic process in our PBPK model. In RFA FD 21-013, we have asked for applications for developing PBPK models to aid in the development of generic dermatological drug products. Our objective is to have a PBPK model that can mechanistically describe skin permeation of both active and inactive ingredients. We also wanted to enable the PBPK model to integrate experimental and in vitro data. In RFA FD 21-018, we have asked for applications to quantify the expression of metabolizing enzymes and transporter proteins in skin tissue. Our objective is to enable PBPK model to account for skin metabolism and or potential transport within the skin with the aim to track down both parent and metabolites. Now let's talk about the use of Dhammal PBPK model in regulatory space. The generic drug developers are now utilizing this in silico methodology to conduct virtual bioequivalence, to define safe space for critical formulation attributes, and to extrapolate bioavailability and bioequivalence assessment from healthy to disease populations. The blue bar in this graph shows the number of regulatory submissions since 2017, where the applicant have provided PBPK model in their applications such as pre-NDA meeting package, and applications, control correspondence, etc. And the orange bar in this graph shows the regulatory task where the applicant did not submit PBPK modeling in their applications, but the agency has developed PBPK model and used it to guide regulatory decisions. The agency also uses PBPK modeling in product-specific guidance development and other regulatory research. We encourage the generic drug developers to explore these unique opportunities. If you are interested to learn more about our research activities, please check out our science and research report on local acting PBPK modeling. Here you would find the summary of our research activities, our research projects and collaborations, and you would also find published articles, posters, and presentations from FDA authors, grantees, and contractors. To summarize my talk today, PBPK model-based virtual bioequivalence is an innovative alternative approach for the approval of Dhammal drug products. Gadufa-funded research is set for continuous improvement of Dhammal PBPK model or modeling platform. Mechanistic Dhammal PBPK modeling is used by the applicants to support their drug development and approval process, and by the agency to make informed regulatory decisions. Academia, industries, software developing companies are encouraged to follow notice of funding opportunity to work with the agency to achieve critical mission of Gadufa. Before I end my talk, I'd like to take the opportunity to thank my colleague, Dr. Eleftheria Tsakalozou, my team lead, Dr. Andrew Babiskin, my team members, and my director, Dr. Zhao, as well as my FDA colleagues. I would be happy to take any questions during the panel discussion session. If you have any follow-up questions, please feel free to reach out to me to this email address listed here. Thank you for your attention and listening to my talk.
Good afternoon, everyone. Thank you so much for joining me today. I'm Priyanka Ghosh. I'm an acting team lead within the Division of Therapeutic Performance, Office of Research and Standards, Office of Generic Drugs. And today I'm going to talk to you about the role of drug product characterization data and drug product performance data towards building a dermal PBPK model. This presentation reflects my views and should not be construed to represent FDA's views or policies. And the learning objectives for us today are to understand the complexity of topical dermatological drug products, discuss some of the bioequivalence approaches that are typically utilized for establishing BE of such drug products, and most importantly, discuss how drug product characterization data as well as performance data that is perhaps already generated within the scope of these bioequivalence approaches can be leveraged and utilized to develop as well as validate PBPK models that can then be utilized for evaluation of bioequivalence. So let's begin by talking about what are topical dermatological products. Well, topical dermatological products encompass a huge range of dosage forms, ranging from solutions to gels to creams to ointments, lotions, foams, and so on and so forth. And the important consideration here is we need to understand not only the dosage form in situ, and what I mean by that is the dosage form that exists in these container closure systems or drug delivery devices, but we also need to understand how or what happens to the dosage form once it's applied to the skin and the drug product is undergoing metamorphosis. And this is critical because understanding the interaction of the dosage form with the skin, especially say, for example, if you have an oil in water emulsion, we need to understand how fast the water evaporates, what does the oil phase comprise of once the water evaporates, and how does that oil phase and the API in that oil phase interact with the skin to contribute towards the overall bioavailability from that drug product. So now that we have talked a little bit about what we need to understand from these dosage forms to build the models, let's look at how have we established BE. Historically, we have utilized either comparative clinical endpoint studies or vasoconstrictor studies for establishing BE. And more recently, we utilize what we call efficient characterization-based approaches, which may comprise of in vitro and or in vivo pharmacokinetic studies for establishing BE. And these characterization-based approaches are designed using a modular and scalable framework where as the complexity of the dosage form increases, we systematically utilize studies to mitigate risks associated with potential failure modes for BE, and thereby the comprehensive data sets allows us to evaluate BE for a given drug product. And such data typically comprises of an assessment of the formulation sameness. We look at physicochemical and structural characterization data, and then we look at performance tests such as in vitro release test, in vitro permeation test, or another biorelevant assay. And in some instances, we also look at PK data. Now, over the next few slides, I'm going to talk you through how do we identify what kind of Q3 data may be relevant for a given dosage form, and how can we leverage that data to then inform a model. So how do we identify Q3 for a given dosage form? So the key consideration here is we know that it is soluble drug in a dosage form that contributes towards the bioavailability of the active ingredient from the drug product. So we need to think about factors that can alter the amount of soluble drug and thereby the thermodynamic activity and the bioavailability of the active ingredient from the drug product. So if we begin by looking at the state of the drug substance, if the drug substance is dissolved, typically we think about what is the pH of the formulation? Is the pH of the formulation close to the pK of the drug, for example? Because if it is, then small differences in the pH of the prospective generic product compared to the reference product could lead to differences in solubility that in turn could lead to differences in bioavailability. Similarly, if there are different isomers for the drug substance, we think about if they contribute to differences in solubility of the active ingredient in the dosage form. Then if we think about dosage forms where the drug substance is not only dissolved, but also there are suspended particles. Here we have to think about, are there any differences between the polymorphic forms of the drug product? Are there any differences in the particle size distribution of the drug substance between the test and the reference product? Because if there are differences in, say, for example, dissolution rate of these particles in the drug product, it could lead to differences in solubility and thereby bioavailability of the active ingredient from the drug product. Then as we start looking at more the formulation, say for example, for single phase gels like a solution or a gel, we think about the rheological profile for these non-Newtonian formulations or the viscosity, because we know that differences in viscosity between a test and a reference product could lead to differences in potentially diffusion of the active ingredient in the dosage form and thereby the bioavailability from the product. And of course, I discussed pH on the last slide as well. For multiphasic formulations like lotions or creams, in addition to looking at rheology, pH, etc., we also need to look at the phases and the arrangement of matter, and specifically one that is easy to grasp is the globule size distribution. Because again, if there are differences in globule size distribution due to perhaps differences in the manufacturing process, that could lead to differences in the bioavailability of the active ingredient from the drug product, especially as the drug product is drying. And then we also try to understand the distribution of the localization of the drug across the multiple phases of the drug product. So now that we have talked a little bit about how do we try to understand what Q3 may be critical for a drug product, what do we typically utilize? Well, this is just an example. For single-phase systems, we start by looking at visual appearance and microscopy data to see if there are any suspended particles. If there are particles, we look at particle size and polymorphic form. We also try to look at drying rate to understand if there are any differences in the rate of metamorphosis of the test product compared to the reference product. We look at density or specific gravity, rheology, and pH. And other relevant Q3 parameters for a given drug product. For multiphasic systems, in addition to the parameters that I talked about in the last slide, we also, of course, look at globule size distribution, and we also look at other parameters such as water activity, etc., depending on the drug product again and the formulation of the drug product. So now that we have talked a little bit about how do we go about identifying Q3, what Q3s are typically relevant for a single phase system compared to a multi-phase system, what do we do with the data? Well, this is an example from our Gadufa regulatory science research projects. We are looking at data from metronidazole products. Specifically, we have the creams on the far left here, and then we have the gels. So you see that we have generated a whole host of Q3 data, including pH, density, particle size is not relevant for this product because the API is dissolved. Globule size is only relevant for the creams, not for the gels, and so on and so forth. We generate this data. We correlate this data with the performance. In this specific example, we used an in vitro permeation test or an IVPT study. And then we look at how the differences in the Q3 parameters, such as, for example, the differences in the drying rate between the cream and the gel, how they contribute to the overall bioavailability from the test product, from the drug products. And in this example, we see that the faster drying gels have much lower bioavailability compared to the more slowly drying creams. And there are, of course, other differences in the Q3 data. Now, the key question here is, if we can generate all of this Q3 data, we can correlate that with performance. Why do we need a model? Well, we need a model because there are unique situations. This is another example from, again, our Gadufa regulatory science funded research. And for the purposes of this illustration, I will use the cream, the lidocaine prilocaine cream here as a reference product and the data from the generic cream as a prospective generic product. Now, if I can draw your attention to the pH data here, you will see that the pH data is relatively close to each other for the for the generic cream compared to the reference cream, but there are differences. Now, typically in our guidances, we recommend that for Q3 characterization, one should evaluate 3 batches of the test product and 3 batches as relevant of the reference product. And then we utilize that data, we utilize that Q3 data to compare the test and the reference product. And usually the expectation is that the values we obtain for the generic product should fall within the range of the values that we obtain for the reference product. And this difference should be similar to what we would expect across multiple batches of the reference product itself. Now, in this instance, you can see that that is not the case. Now, if you were to develop a model here, you have all of the Q3 data. You can use the Q3 data, your understanding of the microstructure of the drug product as it's drying, as well as something like IVPT data or other data sets for local bioavailability that may be available in the literature to develop the model. You can then utilize that model to demonstrate that this difference, this apparently small difference in pH values between the test and the reference product for the purposes of this example, do not impact the bioavailability of the active ingredient from the drug product. And that could be a powerful tool to support your overall demonstration of PE when you observe minor differences in your Q3 parameters. And this is what we mean when we say that you can use a model to establish a safe space around your Q3 parameters. Now, of course, in this example, I talked about using an IVPT data to perhaps inform the model towards both development as well as validation. So I want to take a few minutes of this presentation to discuss how do we generate the local bioavailability data, how do we obtain the local bioavailability data that we can use to both develop as well as verify or validate the models. Now, IVPT studies are recommended in our guidances. You can look into this presentation cited at the bottom of the slide here if you are interested in learning more about the fundamentals of IVPT. My goal here is to not to walk you through IVPT method development, validation, and pivotal studies designs, because of course you will not be designing a pivotal study typically. If you may not be designing a pivotal study, or this may not be of interest to you. What I do want to take this opportunity to do is walk you through how are these IVPT studies designed so that you can appreciate how the models may need to be designed to be able to accommodate such data. In an IVPT study, we typically utilize multiple replicate skin sections from the donor to evaluate both the test and the reference product. We compare the bioavailability of the active ingredient from the test and the reference product within a single donor, and then we compare the data across multiple donors in an effort to understand the overall variability that is expected in the bioavailability of the active ingredients from these drug products. And what we look at typically is cutaneous pharmacokinetic data. So here, instead of concentration, which is typical for traditional pharmacokinetics, we have flux, and then we have time. And we looked at these flux versus time profiles to understand how differences in Q3 perhaps led to differences in metamorphosis of the dosage form, and thereby the bioavailability of the active ingredient from the dosage form. Because of course you see in this example that there are differences in bioavailability between the acyclostat, which is the blue line here, and the Zovirax US, which is the US reference product for acyclovir topical cream. Now, while you may or may not be designing pivotal IVPT studies, it is important to understand parameters that will be relevant during the conduct of these IVPT studies, even for model development and validation. And these include carefully selecting your apparatus, selecting a skin source, selecting your receptor solution, ensuring that your barrier integrity test is actually is able to distinguish skin sections that do not have that have compromised skin barriers. And you also need to carefully select the dose amount, the dosing technique, the dose duration, as well as the study duration, the sampling method, as well as the sampling schedule, to be able to develop an IVPT method that you can then utilize to inform your model. And it is very, very critical to ensure that you are comparing the test and the reference product under identical study conditions, for evaluation of bioavailability. Once you have identified your method parameters, it is also very important to validate your model for sensitivity and selectivity. And this is important because we want to make sure that if you are using data from an IVPT model to inform your PBPK model, that IVPT method needs to be sensitive as well as selective to be able to identify differences in bioavailability if such differences exist between test product and a reference product. And the way to get there is by validating your method and establishing sensitivity and selectivity of your IVPT method, and thereby of the data that you are utilizing to inform the model. Once you have, and this is not only true for an IVPT study, if you elect to use other types of local bioavailability data, such as in this example, we have looked at microdialysis and open flow microperfusion data to measure the rate and extent of bioavailability in vivo in the dermis. You may also elect to use other types of studies or techniques which allow you to assess local bioavailability of the API from the drug product. But the key consideration, again, is to remember that you need to establish sensitivity and selectivity of your method before you use such data to inform both inform as well as to a PBPK model as well as verify a PBPK model. So with that, in summary, I would like to say that topical dermatological products are, of course, complex dosage forms, and it is critical to understand the behavior of the formulation during metamorphosis to be able to develop models that can be then utilized for evaluation of bioavailability. And drug product characterization data, of course, can help you to develop these models. Drug product performance data, both from in vitro as well as in vivo sources, can also be utilized both to develop and validate these models, but you need to make sure that these methods are sensitive and discriminating. And the overall goal of our Gadufa Regulatory Science Research Program is to facilitate the development of these modeling strategies so that they can then be leveraged towards generic drug development. With that, I would like to thank our entire team within the Office of Research and Standards, Dr. Sam Raney, Dr. Markham Luke, Dr. Tanaz Ramadanli, Dr. Megan Kelchin, Dr. Eleftheria Tsakalozou, Dr. Andrew Babiskin, Dr. Lei Zhang, and Dr. Rob Leinberger, as well as all of our research collaborators for their contributions to this presentation and for helping us to develop bioequivalence, characterization-based bioequivalence approaches for establishing BE for these dermatological drug products. I would like to thank all of you as well for your attention, and I look forward to your questions during the Q&A panel. Thank you.
Hello, my name is Jessica Spires. I'm a senior scientist at Simulations Plus, and today I'm going to talk about PBPK modeling of dermal penetration from topical formulations using the GastroPlus software. In particular, today I'd like to talk about physiologically based modeling to support dermal formulation design, and we'll be using the case study of clobetasol to explore this. So absorption of topically applied compounds into the skin is heavily affected by the composition of the topical formulation through attributes such as the solubility of the compound in the formulation, diffusivity of the compound in the formulation, partitioning from the formulation into the skin, and so on. The composition and related attributes of topical formulations, therefore, are going to affect whether the compound is able to reach therapeutic concentrations, whether these are is a compound that's acting locally in the skin or whether it needs to reach the systemic circulation. Okay, and of course, this composition is also then going to be affecting whether or not the test formulations are bioequivalent with a reference formulation. Physiologically based modeling that simulates the mechanisms by which these attributes affect absorption can be used to inform the formulation development by modeling the effect of these attributes, therefore reducing the need for animal and human testing. In GastroPlus, we've developed the transdermal compartmental absorption and transit model. And this is going to be a physiologically based modeling platform used to simulate absorption and disposition of compounds in the skin. It can be used to identify critical formulation qualities and attributes and model their effect on the absorption into the skin and or the uptake into the systemic circulation. By using parameter sensitivity analysis in conjunction with physiologically based modeling through the TCAT model, we can simulate factorial designed experiments to identify the most critical formulation attributes and quantify their effects on the absorption and on the local and or systemic concentrations. We will be looking at the absorption of clobetasol from microemulsions. So at this point, I will briefly discuss the processes we include when modeling emulsions in the TCAT model. We are able, within the vehicle, we are able to model the concentration in the continuous phase, the concentration in the dispersed phase droplets, and also the diffusion as compound moves between the continuous and the dispersed phases. We're also able to account for the dispersed phase droplets being adsorbed to the surface of the skin. So we're able to model the absorption into the skin from both the continuous phase and from these adsorbed dispersed phase droplets. All of these processes are going to be important when considering the absorption of clobetasol from microemulsions into the skin. To go into a little more detail, clobetasol 7-propionate is a topical steroid that's used to treat conditions such as psoriasis. So in this case, we're really going to be most interested in the local concentrations of clobetasol within the skin. When it's applied in formulations such as oil-water microemulsions, the dispersed phase droplets can act as a reservoir for clobetasol so that we're able to maintain drug saturation in the continuous phase and drive absorption. So as a basic workflow, first we will build a baseline model of clobetasol propionate in the skin and its absorption from a specific microemulsion product. Once we validated this model, we can use it to predict and explore the effects of formulation changes on the absorption of clobetasol and concentrations within the skin. So this is the study that we looked at. This was a paper published in 2016. I believe it came from Dr. Frank Senner's lab. And in this case, the clobetasol was applied to the surface of the skin in the form of Dermavate cream, which is an oil-water microemulsion. Okay. Clobetasol concentration was directly measured in the dermis for 24 hours after application, and they were able to measure the local concentration using dermal open flow microperfusion. Okay. So as you can see in the diagram here, they are actually able to directly measure the concentration in the dermis. And in this case, this was at an average probe depth of 0.87 millimeters. So the information on the composition of Dermavate cream was taken from a number of different sources and used to derive formulation parameters. So I won't go into too much detail here. Some of the important ones, obviously knowing the content of clobetasol, its concentration within the formulation, the volume fraction of the dispersed phase. We knew the aqueous solubility and the solubility specifically in the continuous phase of this formulation, and we were able to use these to derive the partition coefficient for the continuous phase. For the dispersed phase partition coefficient, we assumed it was similar to vegetable oil. Okay. The aqueous diffusivity was estimated using our ADMET predictor software based on the structure of clobetasol. And then we were also able to get estimates of the diffusivity in the continuous phase and the dispersed phase. We used standard equations to model the distribution in the skin. We modeled the stratum corneum using the Robinson equation, and we used the Kretzow's equation to describe the permeability in the dermis, and we used the same values for the viable epidermis. So one of the advantages of using the TCAP model is that we don't only calculate the overall concentrations within each layer of the skin. We're also able to divide the skin layers into what we call sublayers according to their depth within the skin. So sublayer number 1 would be the highest one, then sublayer 2, sublayer 3, sublayer 4, and so on. So when we're looking at, for example, this dermal open flow microperfusion probe data, we are able to simulate the concentrations, particularly at the probe depth or at the range of probe depths, rather than only having an overall dermis concentration. So in this case, I believe we divided the dermis into 20 sublayers. And when we did so, sublayers 12 to 16 corresponded to the probe depth of around 0.87 millimeters. Okay, so you can see in this plot we have the CP time profile. The closed circles here are showing the concentration measured by the dermal open flow microperfusion. And then you can see the local concentration simulated at the sublayers of 12, 14, and 16. Okay, so you can see that the DOFM data was matching with this range, and it was closest to the simulated concentrations in sublayer 16. The study also measured the 24-hour AUC at different skin depths, and that's going to be the blue open circles here. Okay, so they put the probe at different depths and measured the 24-hour AUC at these different depths. Okay. So you can see there was quite a bit of scatter in the experimental data, but they did find a statistical decline in the AUC with increasing skin depth. And you can see here with the red crosses our simulated AUC at different probe depths. And you can see that the simulation results pass through the experimental values. So we said this was a good validation of our baseline model. And now we can use this model to start to explore the effects of formulation characteristics on the dermis concentration time profile and on the 24-hour AUC in the dermis. So the first parameter that we explored was the dispersed phase volume fraction. Okay. And in this case, we varied this fraction by plus minus 25% from the baseline value for Dermavate cream. And we chose this range of values because this is the range in which the microemulsion would remain chemically stable. So the effect of increasing the dispersed phase fraction overall decreased the local concentrations in the dermis. Okay. And when we investigated this, the reason was because it increasing the dispersed phase fraction increased the overall clobetasol solubility in the formulation. In turn, this decreases the fractional saturation and reduces the transdermal flux. So in this case, you get less lower concentrations in the skin. The second parameter that we looked at was the dispersed phase partition coefficient. Okay. And in this case, we use this as a way to explore the solubility of clobetasol in the dispersed phase. Okay. So these two attributes will be linked to each other. The dispersed phase partition coefficient increases as solubility increases. And in terms of actual formulation design, the solubility would be varied by changing the hydrophobic excipients. So we looked at the range here from vegetable oil, where you had a dispersed phase of vegetable oil or something similar to it. And then at the high end, we looked at the case where you had a dispersed phase that's similar to octanol. So an increase in the partition coefficient of about tenfold. So as you can see here, the local concentration in the dermis is going to decrease as the dispersed phase partition coefficient and the dispersed phase solubility increase. Okay. And the reason for this is going to be similar to the issue with changing the dispersed phase volume fraction. As this partition coefficient increases, the fraction of saturation in the formulation is reduced, and then in turn that's going to reduce the transdermal flux. So finally, we looked at the combined effects of both of these factors through coupled parameter sensitivity analysis. And in this way, we were able to do a simulation of factorial experiment design. Okay. In this case, we looked at the effect of the 24-hour AUC in the dermis, okay, at the DOFM probe depth. Okay. And you can see here on the x-axis, we have the dispersed phase partition coefficient, as you remember, is going to be coupled with the dispersed phase solubility. Okay. And each of the points here represents different points in our range of dispersed phase volume fractions. So all of the points here would represent different experiments. We're able to derive a response surface for the two parameters that quantifies the sensitivity of AUC of the 24-hour AUC in the dermis to these changes. Okay, and you can see this below. It's going to take this form, and where C1 to C4 are real constants. Okay. And this type of response surface could then be used to determine formulation changes that would allow for bioequivalence between different formulations. So to conclude, physiologically based modeling and simulation of topical and transdermal formulations provides a promising approach to test and predict the effect of formulation changes on drug absorption and distribution in the skin. The sensitivity, using this type of modeling approach, sensitivity to formulation attributes can be assessed for single parameters, and the combined effects of multiple parameters can be quantified through coupled parameter sensitivity analysis. Okay. These approaches can then be used to start to inform the design of generic formulations with the goal of achieving bioequivalence. Okay. While it's not quite a conclusion of this work, I would say that in the future, something else that can be done is to couple these models to virtual population simulations. And this would also be helpful to indicate what type of experimental sample sizes would be needed to detect differences between formulations in a statistically rigorous way. I'd like to thank everyone who worked at the Simulations Plus team on this project. In particular, Bill Van Osdal, Jin Dong, and Yasmina Novakovic, who really worked hard on this case study. This work was performed through a grant with the FDA Office of Generic Drugs, and of course the views that are expressed here do not reflect official policies of the US FDA, the Department of Health and Human Services, or the US government. Thank you.
Good morning, good afternoon. My name is Sebastian Polak from Certera UK, and today I'm going to discuss a case study on how to use modeling and simulation approach for the dermal formulation optimization, clinical development, and regulatory assessment. So I mentioned modeling and simulation, what I had in mind is a PBPK model, where we follow the well-known paradigm, which allows us to dissect the whole problem into the three sub-areas, namely drug-specific data, systems data, and trial design. So when we think about the dermally applied drugs, the drug-specific information include the information about the formulation itself, plus the data which characterize the API itself, like PhysChem data and PBPK information. As for the systems data, we need information which allows us to differentiate between different locations where the drug can be applied, and which can be further extrapolated to different populations. So we need information about the physiology of the and anatomy of the skin. We can't forget about the formulation, which will be the main element of today's discussion. The good model needs to allow to deal with different type of formulations, solutions, emulsions, suspensions, up to the patches. So today I'm going to focus on the solutions, which are deemed to be simple formulations. However, what I'm going to show is that such a simple formulation is not always necessarily that simple. As I mentioned previously, I'm going to discuss this case study based on the multi-phase, multi-layer McDermott model, which is part of SimSip Simulator. And apart from that, I'm going to describe very briefly the IVPT module, which allows you to simulate the IVPT study, which is based on the MPML McDermott model conceptually. Obviously, the combination of these two type of tools, namely IVPT and the ability to simulation of the IVPT study, with the model which describes the whole complexity of the human skin and the human body, in combination with the good quality data which describes the Q1, Q2, and Q3 properties of the topical products, allows you to build a tool for the virtual bioequivalence for complex topical products. So I'm not going to go into the details of the model building. However, I will present a simplified model building plan just to give you a glimpse of how it was done. So there are three main parts, actually. So the first one was the development of the basic model with use of the API characterization. In this case, drug-dependent parameters, PK data plus system dependence data. Then development of the dermal PBPK model with use of the IVPT module. And again, it's a combination of the dermal absorption parameters, which allows or helps to describe the permeation of the drug through the skin, and systems data. Regardless of the level, we can't forget about the verification for verifications. For each of these multiple levels, we need a good quality data sets, which allows us to verify the model which we developed. Once we are happy with the quality of the model, we can utilize this model, for example, for the formulation optimization or virtual bioequivalence study, and that's exactly what I'm going to present very briefly today. So I'm not going to spend much time on this slide. We just went through the literature, collected the data for the nimesulide, which is the anti-inflammatory drug, just to build a simple, or relatively simple, basic PBPK model. And then in the separate search, we found, as we believe, all the information which are available for the clinical studies For nimesulide, in this case, tablets, there is unfortunately no IV data available. And after building this relatively simple basic PBPK model without any optimization or fitting, just by providing the data, we verified whether this model is of acceptable quality for all three studies which I'm presenting here. So it's a Jovanovic study, and the comparison between the observed and simulated data. The same from the Erdogan study. The difference between these two is that Erdogan is a multi-dose study, and only a single concentration time point is measured. So as you can see there, both average and individual data seems to be predicted very well. The third study, which is available, is a Gandini study. There are two different scenarios available here, or data for these two scenarios, namely single dose study and multiple dose study. I'm not presenting any goodness of goodness of prediction metrics, which is not absolutely necessary here. Just a visual check gives us the information that the model is acceptably good. So we can go to the next step, which is the dermal compound development, and this has to include formulation model development as well. So what we need is the information or data, parameters, which allows us to describe the permeation of the drug through the skin. All of them were predicted based on the PhysChem data, as you can see here. But apart from that, we need the information about the formulation. What can be the effect of the formulation on the behavior of the API? In this case, as we believed, the crucial parameter will be the solubility of nimesulide in various organic solvents and mixture of solvents, and that's something that I'm going to use later on. So again, we calculated these parameters, we provided them to SimSip, and then we found two IVPT studies, which we used for the verification of the developed model. The first one is Dial study. In the Dial study, they provided the permeation profiles of nimesulide from three commercially available gels, which were assumed to be solutions, namely Nimolide, Orthoped, and Nisogel. And what we did, we took the data which I described previously and the basic PBPK model, and we run the simulation without any optimization. So here is the prediction. So it is acceptably good prediction actually already, but there is something lacking. There is not necessarily the perfect model, the model which we want to have, which we want to use the formulation optimization of VBE. So the question is, what and how should be optimized or checked or modified to have even better model? As we said previously, and as we know very well, the behavior of the drug is an interplay between the systems data and drug formulation data. So what kind of drug or formulation data can be crucial here? And here, actually, and that's the crucial slide of this whole presentation, because here is the real scientific work. So which of the parameters of all those parameters which are presented previously, which influence the permeation or allows us to calculate the permeation of the drug through the skin, should be measured or optimized or dig deeper to understand its influence on the behavior of the API. So what is absolutely necessary here, and it's a crucial, very useful tool, is a sensitivity analysis, both local and global sensitivity analysis tool, which allows you to understand the influence of certain parameters on the behavior of the API in the skin. There are some hints which we should probably provide. Is parameter of interest should be preferably measurable, and to help to understand the excipient effect and to allow accounting for the various formulations. In this case, after several sensitivity analysis and digging into the results of this analysis, we noted that the partition coefficient between the formulation and stratum corneal lipids is not optimal, because by default, the system uses stratum corneal lipid water partition coefficient, and water does not necessarily reflect properly the complex formulation, which in this case is, as I said, something what is deemed simple, not necessarily that simple. Apart from that, and here's the second crucial information, which is kind of a take-home message actually too, please be careful with the systems data. So what we are using here, as you can see, is the IVPT tool, and still the systems data are important, because depending on the skin, location of the skin from which the skin was harvested, and thus, or apart from that, the gender of the donor, the fit can be very different, and the the value of the optimized parameter can be very different. So in this case, it is absolutely necessary. We tested that, and we know that because of the differences of the male and female abdominal skin, we got different fit for the partition coefficient. So what was done next? We optimized partition coefficient between the stratum corneal lipids and vehicle for these three formulations. Here are the results. But apart from that, and based on the optimized KP, we calculated the vehicle water solubility ratio. So how different is the solubility in water and in the vehicle which was used? And this is actually a relatively simple parameter which can be measured, and in the opposite direction used to calculate the partition coefficient, and thus used to optimize the formulation of interest. The second study was used purely to verify this approach. It's a Pereira study where they used an nimesulide 2% gel with different set of excipients with unknown Q3 parameters. So we didn't know the vehicle water solubility ratio. We noted there is no ethanol present there. So we assume that the solubility ratio will be lower as compared against what we saw for nimolide 1%, for example. So we assumed it will be 4. Then, based on that, we calculated the partition coefficient between the stratum corneal lipids and vehicle, and then we used it to run the prediction. So as you can see, the viable epidermis and dermis amount is predicted perfectly. There is a misprediction as for the stratum corneum. However, it's in a ballpark range, I would say. There was no receptor fluid. There was no drug present in the receptor fluid. We, in the simulation, had a very, very small amount of drug which permeated. Again, it is based on the assumed parameter with further optimization, which was done, and I'm not presenting here. The results can be even better. So we can assume we have relatively good model, which can be taken farther to the third step of the verification. So the simulation of the dermal administration studies with use of the IV-IVE approach. So the previously optimized in the in vitro level parameters are transferred to the in vivo situation, and the in vivo situation is predicted. And here is how it was done. In the Sen-Gupta study, they used nimolide gel, which we know very well from the DIAL study. So we know the vehicle water solubility ratio and thus partition coefficient between the stratum corneal lipids and vehicle. And we used this data to predict the plasma concentration in the virtual population. And the same for the Erdogan study. However, here the situation is slightly different. So the formulation which they used is a sulidine gel, and sulidine has different set of excipients, combination of excipients, and again, we know that there is no ethanol. So we use the same assumptions that we used previously. We assume that the vehicle water solubility ratio is 4, and with that, the calculated partition coefficient is around 18, which was used to run the simulation. And here is the results. So we've got acceptably good prediction of the average. However, when we compare the individual data, we can note that there is one clearly outlier which is present here. So we also analyzed the situation without the outlier, and as you can see, there is a spot-on prediction already. So at this stage, we can agree that we have a acceptably good model which can be used further for the proper utilization of the model. So I'm going to just give you one example of how it can be done. And I'm going to use the virtual bioequivalence strategy for that. So the question, the purely artificial question which we asked ourselves, is nimolide and Nisegel bioavailable? What we've got information about the IVPT study, and we have a good quality in silico model. So what would be the normal approach? We go to the clinic to CRO, we run the PK/PD endpoint study, and we analyze the data and test whether these drugs are bioequivalent. But question is, why not to do it with use of this good model which we have just developed? So what we did, we ran the virtual bioequivalent study in the crossover schema with 25 individuals, and we used plasma and local tissue, in this case dermis concentration, as the endpoint of interest. So what you can see here are the examples of predicted amounts, in this case, of the drug in the dermis after the application of these two different formulations. These data were taken to the Phoenix, analyzed with use of built-in bioequivalence module. And what we have at the end of the day is the result of the virtual bioequivalence study. In this case, what I'm presenting is the plasma concentration. As you can see, the average values of the ratios of average ratios for nimolide and nisogel, and there's 90% confidence intervals fit within the acceptance criteria. When we go to the dermis as a endpoint of interest, we can note that the 90% confidence interval for the Cmax ratios is outside the acceptance criteria. Again, this gives us the information and gives us and proves the ability of good, properly developed PBPK model to run the virtual bioequivalence study, but also to better understand your formulation. Because what we could do at this stage is to take this formulation back to the virtual lab to analyze to what degree we can change the parameter, or we should change the parameter to have bioequivalent drug, and rerun the virtual bioequivalence study to prove the bioequivalence. So this is really a tool which can be used for this kind of the proposals. 3 take-home messages which I want you to take with you. First of all, the physiology matters, even for the simulated IBPD study. It is important. Please don't forget about it. It is important part. Sometimes simple parameters are crucial, and please measure them. Measure a little bit in the vehicle. Measure this a little bit in the continuous phase. Measure the evaporation and the solubility in the after the evaporation. All of them are relatively simple and can really help you define the good quality model, which can be further used to run virtual bioequivalence study, and it can help you to understand your formulation. VBE and virtual formulation optimization are possible once you have a proper tool and good quality model based on the tool. Thank you very much. I'll be happy to take any questions you may have.
Thank you for the very nice introduction. My name is Eleftheria Tsakalozou, and I'm a pharmacologist at the Division of Quantitative Methods and Modeling, Office of Research and Standards, Office of Generic Drugs. Today, I will be discussing with you scientific and regulatory considerations on dermal PBPK modeling for virtual bioequivalence assessments and decision-making. My disclaimer: this presentation reflects the views of the author and should not be construed to represent FDA's views or policies. In the next 15 minutes, I will go over considerations on bioequivalence for dermatological drug products. I will go over a case example of a physiologically based pharmacokinetic model that was used to support the approval of an ANDA. That model was developed for a diclofenac topical gel. And I will use this example to go over considerations on model development, model performance assessment, virtual bioequivalence studies, and reporting and documentation that are relevant to dermal PBPK models. And I will end this presentation providing some conclusions and take-home messages. Modeling skin bioavailability can be a challenging process. It is the result of the complexity in the physiology as well as anatomy of the skin. Skin is comprised of multiple layers. Some of them are vascularized, some of them are not, as you can see on the schematic on the left. The skin is also interrupted by appendages such as hair follicles as well as sweat glands. A skin permeation model should be able to describe the complexity of the skin, and it is often coupled with a full body physiologically based pharmacokinetic model where every organ in the body is represented by a different compartment. Implementation of in silico methodologies for generic dermatological products can be a rather complex process. It involves the integration of information on drug product characteristics, on the skin physiology for individuals as well as populations, and on information on skin bioavailability, which can be extracted for studies such as dermal microdialysis or in vitro permeation testing. All this information is integrated within a computational framework that allows us to predict drug amounts in the skin, the different skin layers, the target site, as well as the systemic circulation. As we become familiar with dermal PBPK modeling, I would like to bring to your attention two recent publications. The first one is an overview of a dermal PBPK model that was developed for diclofenac sodium topical gel 1% and was used to support the approval of an ANDA. The second publication is an overview of scientific and regulatory considerations for dermal PBPK models that are used to support regulatory decision-making. Now I would like to take this opportunity and walk you through an example of a dermal PBPK model that supported the approval of ANDA 211253. This dermal PBPK model was developed for a generic diclofenac sodium topical gel that referenced Voltaren topical gel 1%, and that was the reference product. It was approved on May 16, 2019. And at the time, the product-specific guidance recommendations for this specific product involved a comparative clinical endpoint bioequivalence study and a bioequivalence study with PK endpoints. The alternative B approach involved a test product that was Q1, Q2, Q3 similar to the reference product, and a dermal PBPK model was proposed by the applicant in lieu of an in vivo comparative clinical endpoint B study. An overview of model development, verification, and validation processes is presented in the slide. The dermal PBPK model is composed of a skin permeation model and a systemic disposition model. Both of these contain information on the physicochemical properties as well as the absorption, distribution, metabolism, and excretion characteristics of the active ingredient. The skin permeation model also contains information on the drug product quality attributes. Both of these models, skin permeation as well as systemic disposition model, are verified and validated before being considered fit for purpose. Now let's look in more detail how the previous workflow was applied in the dermal PBPK model that supported ANDA 211253. The model was developed on a commercially available platform. You can see the structure of the model on the schematic on the left. Diclofenac physicochemical properties and ADME properties were taken into consideration when developing the model. Formulation attributes for the reference and the test product, such as viscosity, global size, and pH, were also considered. Inter and intra-subject variability were components of the proposed model. And the applicant modified the deep tissue compartment available within the model to simulate the synovial fluid, which is the presumed site of action for this specific product. Model development was followed by model performance assessment, which was divided into two processes: platform performance assessment and model performance assessment. The applicant developed more than 10 dermal PBPK models for transdermal delivery systems and topical products to support the platform performance assessment process. Multiple doses, product strengths, dosing regimens, populations of different age, and application of products at different anatomical locations were assessed. Systemic and local bioavailability data were used for model performance assessment. And when the performance of these models was considered satisfactory, the platform was deemed suitably validated. The next step was to move to model performance assessment. The models for the diclofenac topical products were verified, where the code was reviewed and the biological plausibility of the models was examined. Model validation involved dermal PBPK models for diclofenac sodium topical products, solutions, gels, and emulsions, and literature and application data on different doses, different product strengths, dosing regimens, routes of administration, and local and systemic exposure data were used for that task. The next step was to develop dermal PBPK models for reference and test products. The agency refined the applicant's model to improve local exposure predictions by accounting for protein binding in all skin layers, by updating drug product quality attributes, and by modifying certain partition coefficients. As shown in the schematic on the left, the performance of the refined model, shown in blue, is much improved compared to the performance of the model that was originally proposed by the applicant, shown in red. Panels A, B, and C are predictions in the subcutaneous. Panel D are predictions in the muscle, and panel E are predictions in the presumed site of action, the synovial fluid. Validation was performed leveraging macrodialysis and skin biopsy data. Model structural and numerical identifiability was considered. And when the model performance was considered satisfactory, the overall model was deemed suitably validated. It is evident from the example that we just went over that the verification and validation methodology supporting fit-for-purpose dermal PBPK models is a multi-level process. It involves the validation of the platform, as well as the verification and validation of models that are developed for dermatological products and dosage forms of the same drug substance as the reference and the test product, and of the validation and verification of models that are developed specifically for the reference and the test product. When all of these are validated and verified, the models for the reference and the test product can be used for a virtual bioequivalence assessment. This is a slide providing an overview of the implemented virtual bioequivalence workflow. The workflow begins with a verified, validated model. So in the case of the diclofenac dermal PBPK model, that is a model that provides predictions for the reference product seen on the left and the test product seen on the right. Predictions shown with the blue lines agree well with the observed data shown with the red circles. This validated, verified model is then used to conduct an in vivo virtual study. Predictions for the reference and the test product are generated in the plasma, seen on the left, and the synovial fluid, seen on the right, that is the presumed site of action. These simulated PK profiles are then used to conduct a bioequivalence assessment where PK matrices such as the maximum concentration and the area under the concentration versus time curve are estimated for the reference and the test product and compared following statistical tests. Now let's go over some key considerations in implementing a virtual bioequivalence assessment. A virtual bioequivalence assessment is essentially a comparison of simulated PK profiles between the reference and the test drug product. This comparison can take place at the application site and the different skin layers, or at the site of pharmacological action, the target site. In the case of diclofenac, that will be the dermis and the synovial fluid. The comparison can also take place in the systemic circulation, such as the plasma in this case. Accounting for all sources of intra and inter-subject variability is extremely important within the context of a virtual bioequivalence assessment. These sources of variability can be skin physiology parameters, application sites, characteristics of the virtual population, as well as variability around drug product characteristics and how they impact local bioavailability. PK matrices are used for B statistical analysis, and these may be the maximum concentration or the maximum amount, or the area under the concentration versus time curve. The overall shape of the PK curve, including the time to maximum concentration, as well as the absorption and elimination phases, are considered during model validation. Applicants are encouraged to follow best practices when developing dermal PBPK models for regulatory submissions. These are summarized in the guidances issued by the FDA. The one on the left it refers to format and content for physiologically based pharmacokinetic analysis, and it was issued by the Office of Clinical Pharmacology. The one on the right is a rather recent guidance on the use of physiologically based pharmacokinetic analysis for biopharmaceutics applications for oral drug product development, manufacturing changes, and controls. Our take-home messages. PBPK models for dermatological drug products can be used to support development of a drug product prior to its approval. Alternative B approaches for drug product approval. Model development is an intense and resource-demanding process. These models are complex, and so are the drug products that they are describing. There are limitations in data availability in model development and validation. We do support early interaction between industry and the FDA, and we believe that the pre-NDA meeting request program under Gadufa 2 is an excellent platform for that. All the research grants on dermal PBPK modeling that have been funded under the Generic Drug User Fee Amendments 1 and 2 are listed on this slide. If you're interested to learn more about our regulatory science program, please follow the link at the bottom of the slide. I would like to end this talk by acknowledging the contributions of my DQMM colleagues, Dr. Babinski, Dr. Alam, Dr. Walenga, Dr. Tan, Dr. Fang, and Dr. Zhao. The contributions of Dr. Ghosh, Ramanzali, New, and Dr. Luk from our sister division, Division of Therapeutic Performance 1, and very significant contribution and guidance from our immediate office, Dr. Lei Zhang, Dr. Robert Lamberger, and Dr. Sam Raney. Thank you very much for your attention. I hope that you have enjoyed this talk. I will be very happy to answer any questions you might have during the panel discussion session. Thank you.
Thank you to all the speakers from session 2, and we're just getting ready for the panel. So if you have any questions, please post them in the chat box so the panel can address your questions. Thank you.
All right. Welcome everyone to the panel discussion of this session. I'm Sumit, Arora. I hope you can all hear me. If you can just quickly confirm that. All right. I'm Sumit Arora. I will be moderating this session along with my co-moderator, Dr. Sam Raney from the Office of Generic Drugs at FDA. First and foremost, I would really like to thank all the speakers for their excellent presentations and all the hard work which went in preparing these recordings. In addition to our speakers, which are serving as a panelist today, we also have a number of other panelists, which I would like to quickly introduce. So if I may request that when I pronounce your name, you can just quickly raise a hand so then the audience know who we are introducing to. So to start with, I'll introduce Dr. Ping Zhao. Dr. Ping Zhao is currently a senior program officer at Bill and Melinda Gates Foundation, where he manages multiple investments with the goal of routinely applying quantitative methods, including PBPK in global health. Before joining the foundation in 2017, he worked at FDA as a reviewer, primarily in the area of PBPK modeling. Glad to have you with us, Dr. Ping. I would also like to welcome Dr. Lanyan Fang. Dr. Fang currently serves as acting deputy director and has served as associate director of the Division of Quantitative Methods and Modeling, Office of Research and Standards since February 2019. Prior to that, she served as team lead of the Quantitative Clinical Pharmacology team within DQMM for 5 years. She has established herself as an FDA expert in the use of quantitative clinical pharmacology approaches in the review and regulation of generic drugs. She co-leads CEDAR work group tasked with the use of partial area under the curve for bioequivalence assessment. Welcome, Dr. Fang. And lastly, I would like to welcome Dr. Srinivasa Sammeta. Dr. Sammeta is currently working as an associate director product development at Teva Pharmaceuticals, Utah. Dr. Sammeta has extensive experience in formulation development of topical, transdermal, and ophthalmic dosage forms and in vitro permeation testing studies. Thank you, Dr. Sammeta, for joining this panel. And with that, over to you, Sam.
Thank you, Sumit. And thank you so much to all our presenters and also to you, our audience who are attending today. It's really a pleasure to have you with us because we feel that we are at a really critical point in time where we are making a transition from what had been, I think, in the past, this sense that is modeling and simulation really ready to change drug development paradigms. And what I mean by that is a lot of study and research over the decades has gone into trying to figure out what the right approach is for building the infrastructure within a generic drug company, or for that matter, a new drug company, of how you even approach the development of that drug product and how that can impact your timelines and the success and the risks. And I think we're now at a place where it is extremely clear that for generic drugs, that modeling and simulation has a key role to play and really is just very smart and necessary to incorporate into all drug development paradigms. And so the idea that, well, there's these kind of isolated uses for modeling and simulation, I just don't think that's true. And I think we've demonstrated that, and at least we're hoping to be able to explain that as part of this session. One of the concerns had been, well, where in my drug development paradigm am I going to use this? Am I going to use this to help actual, candidate selection or product development? Yes, absolutely. Am I going to use this to actually support a demonstration of bioequivalence? Yes, absolutely. We've demonstrated that it's absolutely feasible to be able to do these, that the infrastructure to describe how you do this, the models are well developed now. And so then part of the question was also, well, where am I going to get the information to put into the models? Well, I hope what you've also seen during the course of this session is that that information is information that you are already collecting as part of your characterization of your product and of the reference product through the drug development process. So most of this information you're already going to have. It's not a huge lift to take that information and now start working with modeling and simulation, in many cases using platforms that are now highly advanced for supporting specifically those purposes. So then the final question might be, okay, I have the information, I can do this, but is the agency going to accept it? As you saw in Dr. Khondoker alum's talk, the agency is using it even more than industry is for all the applications that he had on his graph there. We have such faith in this science. We are compelled by it. We have been studying and researching it for so many years now that we are using it to support regulatory decision making, even in situations when the applicant has not suggested that. And so I hope that what we can do in the course of this panel discussion is figure out how do we now reach this inflection point where this is now just a standard part of the drug development paradigm for generic drugs. And I want to begin by maybe asking the panelists some general questions. Clearly, FDA strongly believes that modeling can be used to complete a compelling demonstration of bioequivalence in numerous contexts, but especially where it's simply not feasible for empirical evidence alone to be able to demonstrate this. So, and I wonder if you can speak to how do we move from putting out guiding principles for doing this to actually specific recommendations for how you would do this for specific products, to actually generalized recommendations for how you do this in general. I wonder, Dr. Fang or Dr. Zhang, if you would be interested in kind of commenting on this.
Okay, I thank you, Sam. I will start by sharing that. So today we heard that thermal PBPK is really a tool to support drug development. So it's really up to the applicant to determine if the thermal PBPK can create a value or can be used to support the approach they want to demonstrate bioequivalence, regardless if thermal PBPK is recommended in the PSU or not. So regarding the specific recommendation on a specific drug product, and if the applicants have any specific questions, I think the message you heard today is you're welcome to contact FDA through controlled correspondence or pre-ENDA meeting package. We are more than happy to discuss with you and to provide feedback from there.
Ping, would you like to add to that at all?
Can you hear me okay?
Yes.
Yeah, great. Yeah, thank you, Dr. Raney and Dr. Fang. This is definitely a moment where I concur 100% or 120% with your comments on the turning point. I feel like while I was listening to you with the opening remark on this turning point notion. Just I got like a goosebump on this, especially I was the one being the person experienced the early days in the agency, looking at the novel technology and how to kind of like turn that into a kind of a industrialized tool in drug development and discovery. And then definitely Dr. Fang is coming in with a bit of humbleness, right? It's like it's up to the sponsor. So I get that. But I have to say it's a really encouraging point where we can make the change. There are a couple of directions that we can do this. I think the driving force number one is we have a lot of experience already, right? It's not like we're looking at Dermal specifically, apply the PBPK model for the first time. And we can leverage a lot of learnings from other applications. Using another term would be the context of use, right? If we refine or define the narrower scope of the use case, there are a lot of potential for the existing platforms to shine, right? And across different disciplines, across different applications, if you look at Dr. Huang's presentation earlier in the morning, the experience accumulated in OCP would give us a lot of confidence to kind of pave our way here for this particular application. So that's one driving force. The second driving force I feel very kind of encouraging is over the last, I don't know, 10 years probably with the Gadufa 1, 2, or maybe 3, you guys did a lot of investments, right? So there are, look at all these platforms being updated within SimSip, within GastroPlus. Now that I'm out of the agency, I can speak more freely about the name of the software products. But that's the reality, right? So we have the technology moving so advanced that there's no reason for us to kind of hold back and say, oh, are they really ready? Or I think for certain use cases, they are ready. There's just no doubt about it. Now it's just a matter of, how we collectively, drive the agenda even further. Now there are a couple of ways we can do it, right? So we can do through, like updating the guidance, right? Put a guidance out, even if it's draft, kind of letting the world know that, hey, you know, we're looking at all these kind of in a day in, day out fashion. And here's the experience over the last 3 years, you know, the number of cases being evaluated internally and the number of cases we're seeing submitted to the agency. So it's a new norm, right? So I think the guidance development will be another critical kind of a driving force behind this, and also publishing this, right? So Dr. Allen, you know, for the case studies, let's find a way to kind of publish in a peer-reviewed journal or publish in you know, like a internal database, or like make the internal database explicit, right? You know, with an anonymous way to kind of hide the sponsor's name if you know, it's too sensitive at the moment. So I think that way you're putting out a gesture that you know, you have the high confidence, right? The agency has a high confidence. I think that would really drive the science. Now, are we being too kind of optimistic? I don't think so. Again, there are tons of experience in PBPK in the last 10 years, right? So there's no need to, I mean, definitely the science needs to be updated, right? So that's why I know we'll get into the discussion around the variability, and there's still lots of things need to be updated. Lots of gaps need to be bridged. But aren't we have a solid sort of a structure model already for representing the thermal physiology? I would say yes, right? You look at what Dr. Polak's presentation and Jessica's presentation. I feel like, you know, how much better do we want this model to be, right? So I'll just stop here. I tend to speak a lot on this topic. So.
Well, thank you. And are there any other comments from the panel relating to this issue of, you know, we've put publications out there, we've put a lot of presentations and workshops and sessions where we've talked about guiding principles. You know, I think one of the usual progression is that product-specific guidances describe specific implementations, and then once you've got, you know, a lot of product-specific guidances and you have a lot of experience with specific implementations, then it makes it easier to generalize. One of the challenges, though, with, you know, calling out an approach in a specific for a specific product is that it may seem to suggest that somehow it's not applicable to all products. And I think, you know, the position right now is this is equally applicable to all products. It really depends on the problem you're trying to solve. I want to move on to some other topics too, but just any final thoughts on that from the rest of the panel?
Yeah, just I agree with Dr. Zhao's comment on having a general draft guidance to let the world know that this can be used as an alternative approach. Because I feel like once that is out, there could be a lot more products that would be bought into the generic space, which are not taken up because of the complexity of the studies involved and the timelines involved. So I agree with that suggestion.
Okay, thank you. Thank you.
Yeah,
so one of the other things,
probably just quickly add, Dr. Raney, I think the putting a general guidance out, the question is always, is it mature now, right? But just one fact I would like to bring up is if you look at the presentations prepared by 5 speakers, I don't think they do a lot of communication about it. I'm going to present this. Are you going to, you know, be okay with that? There is a common kind of a workflow that is very critical. Is being kind of a similarly presented, right? There's some variation about like how this modeling should be done from data collection to, you know, validation, or like, you know, like the verification, and then demonstrating the performance and all that before you apply for VBE. I think, I mean, the way I look at it is pretty similar. So meaning that there is some sort of a consensus, at least within, you know, this virtual panel, with colleagues coming from academic, from industry, from regulatory, based on their experience. I think that's a very, very powerful kind of evidence to drive this first draft guidance. I'm going to stop here.
Well, that's really helpful feedback. That's the purpose of this this session. So thank you. That's really fantastic. So one of the other things that you had spoken about was, you know, variability, and so as we think about you know, in order kind of for a mechanistic PBPK model to be used in a virtual demonstration of bioequivalence, you need to adequately account for the inter and intra-subject variability. And I think that Dr. Tsakalozou did a really nice job in her presentation of kind of discussing the considerations related to population variability. I'd be interested to hear Dr. Spires and Dr. Polak about if you can explain a little bit how this inter and intra-subject variability has been implemented in your respective modeling platforms, in terms both about how you characterize the variability experimentally, and then how you actually implemented that into your models. Jessica, would you like to maybe comment first?
So within GastroPlus, we have an option to do what we call population simulations, where you're essentially, sort of creating and generating sort of, virtual subjects, and their different qualities are kind of stochastically varied. So currently we do include the dermal model with that. Some of the things that we would be looking at with that would be, variations in the thicknesses of the different skin layers and the hair follicle density, all of these physiological aspects that might be different between different people. This is also something that we're looking at very closely as well to see, how well can we start to expand this and start to really get a good idea of the physiological variability between skin and different people and things like that. So, so this is definitely something that we're interested in. We're able to model this type of variation, but we are, actively working on making it more, kind of, more physiologically based, what the variation would be. And the other aspect that we think about as well is, of course, there's variation in the skin in different regions in the same subject. So in that case, we're able to take that into account.
Okay, thank you. Sebastian?
Yes, thank you, Sam. This is something what is absolutely crucial for each and every PBPK model. That's something what I presented at the very beginning as a introduction to the presentation, actually. So between subject variability is something what we know relatively well, and even if there are some elements which are not perfectly covered, that's something what I think I'm probably brave here. I would say we know how to do that. Intra-subject variability is of interest as well. So we've got up to 8 different locations at the moment. All of them are fully parameterized based on the data which come from the literature and from the cooperating universities. We had a FDA grant together with University of Queensland. So that's exactly what we are working on, what we focused on. But what we should also discuss, I think, is between occasion variability or inter-occasion variability. And that's something what is most challenging at the moment, I think. And it's not that easy to parameterize that. That's something what is part of the virtual bioequivalence system, which we have in SimSip. And among all the parameters which we believe can change between occasions, we do account for at least a couple of them, which is, I think, good beginning. And again, that's something what we should remember about and try to find data and maybe even try to find some funds to pay for studies to get this data. No pressure, Sam.
Well, you talked about the parameterization of the model. So let me ask, where do you see observed data? When anyone who's doing this would see kind of observed variability, I'd be interested, how does one deconvolute that observed variability to figure out what the relative contribution is of any of the kind of any individual physiological factor, and how these different physiological factors interact as kind of contributing to among different sources of variability?
Yeah, so this deconvolution actually is done by the model itself. So once the model is properly parameterized with the data which come from the large studies from a relatively large group of individuals which were tested to provide some physiological parameters, this deconvolution is done automatically. Whenever there is any doubt regarding the influence or the importance of certain parameter, sensitivity analysis is a tool which can help to understand that further, and even show whether there is any missing parameter or we do not account for something. In such a case, sensitivity analysis is of crucial importance. There is one thing, though, which I really want to underline here. For the IVPT study simulation, we do have to account for that as well. So IVPT studies are prone to have relatively large variability, and this variability does not necessarily come only from the, let's say, lab-specific sources. They can come from the physiology-specific sources. So that's something that we have to remember about, and we do have to account for that.
Fantastic. Thoughts from other panelists related to variability and kind of the contributions of individual physiological factors?
I think I just would like to add one quick point, and then over to you, Eleftheria. I think it's good to understand that variability a bit, but what we also need to understand a bit is how one parameter within a PBPK model relates to another. So for instance, if somebody has large stratum corneum or thicker stratum corneum, how does that relates to epidermis and dermis? Do we understand that covariation? Because the power of PBPK is good that PBPK can explain the clinical data, but the major power and the benefit of PBPK is it's in prospective simulations. And if we want to really do that prospective simulations, we do need to understand how we can generate these virtual subjects realistically. So do we understand that covariations? How does one parameter is related to another?
I agree. I agree completely. Eleftheria, did you want to comment on that as well?
Yeah, I wanted to provide a very short comment that I think that's a key question. Your original question, Sam, is key because there are a lot of parameters that come into play when we look at the data and when we look at observed data. So we need to go back and look at the sources of variability. What are they? In every single case, and they can be physiology related, skin physiology related. They can be also attributed to the methodology that is used for measurement. So that's another parameter, and the methodologies that we are using may not be as precise as we would like them to be. And then also there's variability from subject to subject, and then there's variability within the same subject. So in order to be able to tease out all these different sources of variability, we need to be able to design studies in the right way to be able to get to the right answers. And these studies at the beginning may have to be short scale, small samples, but then these will eventually accumulate to larger data sets. And then we also need to potentially accept that part of that variability will not be, cannot be attributed to any specific source. So I agree that modeling is an important tool in sensitivity analysis. I'm 100% in agreement with Sebastian, but I think observed data is important as well, should be our starting point, because a model can be misspecified, for instance. So then sensitivity analysis results will not be reliable. So we need to be aware of all these considerations. Thank you.
Thank you so much. Any other thoughts on that? So then let me ask, what are the thoughts from the panel about how do you know when you've built a suitable model, one that adequately incorporates this kind of variability, for example, for a given purpose? Dr. Zhao, do you have any thoughts on that?
Yeah, I was just about to follow on the discussion that Absolutely. Let me see. I always butcher your name. Sorry about that. Eleftheria, about your comments on the variability. I think I hear the concern, and I think this will never go away. It not only for this thermal PBPK. I think the in new drug development, we continue to kind of I shouldn't say suffer from this question, but with just like a KCO2 measurement, right? With the PGP inhibition, with the CYP3 inhibition, with like a FM of a substrate drug for DDI, there are always like different sources of variability. My point is, going back to our earlier question around the sort of generalizability and where we are, whether we feel confident about pushing this agenda using PBPK. I just want to make sure that we don't use the variability to kind of hamper our progression. One thing to kind of throw out there, I do not have a solution, but if you look at the chat, right? Look at the chat box. People were asking, oh, Sebastian, did you incorporate like fatty skin, or like the psoriasis skin? I think at that level of let's call it deterministic differences, right? If the model is not accurately accounting for those disease-related difference, and that can be just the mean population psoriasis versus mean healthy, right? If we don't even understand that difference, I mean, just try to stretch to kind of incorporate any kind of random variability, to me is not useful, right? But if you have just at the level of mean differences, for example, we encounter that very often in new drug when we do drug interaction. Let's say if you have a population that is known to have a GFR that is representing the severe renal impairment, and that mean level of difference will be adequate to kind of help you make a very sound decision. We're not saying that we're able to predict spot on the next patient how his or her PK will look like, but in general, the subpopulation, your simulation will be really, really useful for that scenario. So I think that's really the strength, going back to Dr. Arora's comment, that we always hope that PBPK will do the slam dunk, give us the right answer. But the fact that you see all your internal kind of uses higher than the submission, I think that's that speaks to the recognition by your team to utilize these models, because it does help you to kind of like think through the problems and make the right decision. It doesn't have to be a dose decision, right? I think a dose decision is the ultimate goal, whether we can leave the study, do the dose decision. But yeah, so
Dr. Fang, did you have a comment as well?
Yes, I really want to echo what Ping just said. We have to come back to the purpose of our thermal PBPK for bioequivalence assessment. So if the fundamental question is really to detect formulation difference, if that's our fundamental objective, so I think the focus, we probably, the variability is absolutely important, but at the same time, it's the model, the predictive capability. Can the model reflect the PK change in response to formulation changes? I think that, to me, that's kind of the first level of the question we need to assess in terms of model performance. Thank you.
Thank you. So, I would actually love to spend a little bit more time, hearing thoughts from the panel about how a prospective drug developer can approach this during their drug development program. But I also want to make sure we get to some of the questions from the chat. So I wonder, Dr. Arora, would you be able to maybe lead us through some of those questions, and then we'll see how we're doing for time?
Sure. Let's deep dive into some of the questions we are getting. I think some of them are quite interesting. So one of them is, are increased sunscreen? One of the question is from Nazima. Are sunscreen products combinations as well as standalone products evaluated? So maybe Priyanka, if you want to take that.
Yeah, absolutely. Thank you, Sumit. And thank you for the question. I think I'll begin by saying that most of the sunscreen products are not approved under NDAs. So they are a little bit beyond the scope of the discussions that we are having today. But I also, from a scientific perspective, we do think that potentially some of the in vitro tools as well as some of the in silico tools that we are discussing within this session may be relevant for sunscreens as well to assess bioavailability, given the current interest in understanding the bioavailability from these sunscreen formulations. And especially, throughout the life cycle of these sunscreen products, we often see that there are formulation changes due to availability or inavailability of certain excipients and so on and so forth. And in those situations, the models may be very helpful. They may also be helpful to help to design the must studies that are often required to support the approval of these products, where you are trying to design maximal usage conditions or identify maximal usage study conditions. And in those situations, model predictions can be helpful. They can also be helpful to bridge back to such must studies for the reference product or the original formulation for the sunscreen when additional formulation changes are being incorporated during the life cycle of the product. So scientifically, yes. However, from a regulatory perspective, sunscreen formulations are a little bit beyond the scope of what we are discussing today.
Thank you, Dr. Ghosh. There is another interesting question from Andres Fresquas. I hope I pronounce your name correctly. Maybe not. Does your mathematical model represents the morphology of the stratum corneum and discerns the penetration of the drug and excipients along the laminas in the stratum corneum?
I think that one was addressed to me originally. So in the GastroPlus model that we have, we aren't completely mechanistically modeling all of these types of processes. Essentially what we have is for a layer like the stratum corneum, the primary parameters that we're looking at will be the diffusivity, describing the diffusion within the layer, and then the partition coefficient, which is also describing how the partitioning occurs between the different layers. Having said that, within the standard equations that we use, most of them are going to be accounting for these different processes and kind of boiling them down to a single number. So in the case example, in this clobetasol example, for example, we use the Robinson equation, and with the Robinson equation, it calculates the permeation for the lipid fraction and the protein content of the stratum corneum, and then those are put together. So I think, in the cases where you really are interested in what's happening in these, some of these specific mechanisms, it can be very fruitful to look back and look at the sources of the equations, to see where you can modify different parameters or view different parameters to see what the effect is.
Thank you. Thank you, Dr. Spires. There is another question for you, Sebastian. How do you represent the morphology? And it's again coming back to the disease skin. How do we model psoriatic skin in the model?
And I guess this question was again from Andrzej Przekwas. It's so simple to pronounce it. Andrzej Przekwas. Come on, Sumit. So Andrzej, great question. Thank you. So the disease skin is something important from the practical point of view, obviously. Psoriasis is a systemic disease. So all the changes which we can expect are not only skin specific, they can be internal organ specific as well. But what I'm going to focus during this answer on the skin specific parameters. So there are multiple of them actually, and they should be taken under consideration when you're simulating the disease population. So beginning from the surface of the skin, pH is different. It's lower in general as compared against the healthy skin. The temperature changes. Stratum corneum is different, obviously. Apart from the modified thickness, we've got cracks which can reach down to the viable epidermis. So that's something what we have to account for, and we do. Thickness of deeper layers of the skin, like viable epidermis and dermis, and probably something what is important from the drug clearance perspective is blood flow. So the blood flow is increased as compared against the healthy individuals. So all of these parameters should be considered, and they are considered as part of the population description in SimSip. I hope it answers this question.
Thank you, Sebastian. And maybe a quick question for you, Dr. Alam. FDA has awarded many grants over the years, and you nicely show that list to us. How does FDA determine the research needs that deem necessary for these continuous improvement of these mechanistic PBPK models?
Yeah, thank you. Yeah, so it's a very nice question. So, well, our knowledge on skin physiology, role of formulation, or overall Dhammal PBPK model is evolving over the years. So, the research needs we are identifying today is based on the knowledge we are gaining through the internal and external research, and by reviewing the applications like ANDA, PNDA, or control correspondence, or other sorts of regulatory tasks. So we are collecting these ideas and publishing these research priorities. So you can find these research priorities by searching the word generic drug research priorities and projects. But yeah, I want to also mention here that it is also important to understand that certain research need cannot be executed once the prior research need is executed and successfully accomplished. For example, when our model is capable to predict the permeation of API from a simple formulation, then we can think of how we can incorporate the role of excipients or inactive ingredients in the permeation of API from the more complex formulation. So these are continuous process. So once as we go forward, so we identify new research need, and we try to accomplish through our Gadufa fund.
Anybody else want to further comment on it? Maybe Dr. Raney?
Well, actually, I'd love to hear from Srinivasa. We haven't had a chance to hear from you. Do you have some thoughts?
Yeah, I think that's a good resource. And from industry perspective, like I personally go to the research initiatives and look at what's going on and what FDA is looking at newer techniques to approve or help in the AND applications. And that's very helpful, like the IVPTs and also the PBPK. So I say it's a very good, useful resource, useful for the generic industry.
Good.
Oh, great. I don't know how we are doing on time, but I also see a very interesting discussion going on in the chat as well. So I would really refer everyone to read it. Some really excellent points Amin and Rob is making there. So are we okay with time? We keep going for a certain while, or how are we doing?
We probably have time for one additional question, Dr. Arora.
All right. So with that, I would probably take one of these questions from probably Dr. Eleftheria can comment on that from a regulatory standpoint. What are the specific considerations that applied on the use of PBPK modeling and simulations towards the support towards supporting the approval of ANDA 211253?
Yeah, thank you for this question, Shamit. So when it comes to the approval of the ANDA 211253, I think it's important to keep in mind that the modeling and simulation approach contributed to a totality of evidence approach that led to the approval of the ANDA. So the applicant showed in the NDA, the applicant showed Q1, Q2, Q3 similarity between their test product and the reference, and they had a passing IVRT and a passing in vivo PK study. And what they showed with their model, they established a link between their the systemic bioavailability of diclofenac, sodium diclofenac, which was measured with what is happening in the skin, in the presumed site of action. So that was the link that was missing, and that was the role that the verified and validated dermal PBPK model played. And by establishing that link with a model that increased the agency's confidence that the reference and the test products are bioequivalent at the presumed site of action. So the model contributed to the totality of the evidence that led to the approval of the ANDA.
Thank you, Dr. Eleftheria. Anybody, any final comments from anyone on the panel?
I would maybe just offer one final comment to any prospective generic drug developer who's experiencing some challenges related to the development of a complex generic. I strongly encourage you to consider whether modeling can help you resolve that challenge. Because if you haven't considered that, then there's a very good likelihood that the answer is yes. And then it's just a question of, well, how do I do that? And there are a lot of resources to help you approach that, including a lot of resources that FDA has developed. Lucy, I don't know if you'd like to comment on that at all before we wrap up. I think you may be on mute, Lucy.
Oh, yes. Yeah, thank you, Sam. I want to comment on the question on the research priority. I want to say we really want to engage our industry colleagues in defining the research priority. That's why we have the annual regulatory science meeting every year, so we can solicit your feedback, your input in terms what research can address your pinpoints during your drug development. So we really want to work together with you in defining those research priorities. So thank you.
Great. So thank you, really thank you, everyone. I think we had some really fantastic panel discussions and such warm call out there from our FDA colleagues and all the great work and all the resources which have been went in getting us where we are today. I think it's really encouraging, and so I just really want to thank all the panelists for great discussions, but also you, all the audiences who are staying with us and helped us in giving us some really great questions to get this discussion ongoing. And with that, I would like to thank everybody on this call today. Thank you, and have a nice evening.
Thank you so much.
Thank you,
so
Dr.
much.
Arora.
Thank you.
I'd like to thank our panelists, speakers, and moderators from session 2. That was such a great session. And as Dr. Arora pointed out, there is a great discussion going on in the chat panel. So if you haven't already seen that, please take some time to review that as well. And if you have any additional questions, please do continue to post them on the chat window. We will break for 15 minutes now. We are at 3:02. Actually, we'll come back at 3:15. So 15 minutes, and we'll get back for our session on long-acting generic products.